Now, there are different roles of transcription factors which are played during the protein synthesis by the cells. And in this segment, we will discuss the roles of transcription factor which they influence on the transcription mechanism. Now, transcription level control, that is, the transcription level control is arranged by a large number of proteins. Remember, the proteins are the major working horses for controlling the transcription of the different signals. And these proteins which control the transcription level, they are called transcription factor, of course. And these proteins can be divided into two functional classes. There are two functional classes which control the transcription levels. And the one is the general transcription factor that binds at the core promoter sites in association with RNA polymerase. This is the one factor. And the second one is the sequence-specific transcription factor. These are the factors which are specific to certain sequences which make the protein. And these, they bind to the various regulatory site of a particular gene. The different particular gene, what these do? Sequence-specific transcription factors, they bind to the regulatory sites of particular genes. Now, sequence-specific transcription factor that can act either as transcriptional activator and that stimulates the transcription of the adjacent gene, or as a transcriptional repressor that inhibits the transcription. So it means eventually that the sequence-specific transcription factor, which are very important, they act either as transcriptional activator, that is, they can activate the transcription which stimulate the transcription, of course, or whatever, they can be another transcriptional repressor, that is, they repress, they inhibit the transcription of the gene in this area. Now, a single gene may be controlled by many different DNA regulatory sites. Remember, this is an important fact, that a single gene can be controlled by many different DNA regulatory sites that bind a variety of different transcriptional factors. Now, transcriptional factor, there are a large number of transcriptional factors, and a single gene may be controlled by this. But conversely, a single transcriptional factor may become attached to numerous sites which are controlling the genome, thereby controlling the expression of the host of different genes. So, the one fact to remember that a single gene may be controlled by many different DNA regulatory sites, but a single transcription factor may become attached to numerous sites around the genome and vice versa. The role of transcription factor in determining a cell's phenotype is very important. And embryonic stem cells, that is the ES, they appear very early in the development of a mammalian embryo. We know this. And they are the key properties of these cells are, one is they are capable of indefinite self-renewal. The embryonic stem cells, they are capable of indefinite self-renewal process. And the second is the role of transcription factor in determining a cell's phenotype again is that the embryonic stem cells ES, they appear very early in the development of a mammalian embryo, and their key properties are, one is they are pluripotent. That is, they are capable of differentiating into the all of the different types of cells in the body. Pluripotent, for example, remember, from one fertilized egg or fertilized from stem cell, it is capable of developing into different organs, differentiating into different organs. It can become an eye, it can become a heart, it can become bones, and so on. So this is one of their very important property. Now, these pluripotent stem cells, they have a particular set of transcription factor that played an important role in maintaining their self-renewing and their pluripotent state. This is that how these pluripotent stem cells, they behave like this. That is, they have important role in this because they have particular set of transcriptional factors, and those transcriptional factors, they make them possible for self-renewing and a pluripotent state, that is, they can develop or differentiate into the different types of cells. Now, structure of TFS or the transcription factor motifs, that is the DNA binding domains of most transcription factor, they can be grouped into several broad classes. They can be grouped into several different classes, and among the most common motifs that occur in the eukaryotic DNA binding proteins are the zinc finger, the helix loop helix, and the leucine zipper. These are the three types of the motifs which are the transcription factor there. And what is the zinc factor motif? That is the largest class of mammalian transcription factor. It contains a motif which is known as zinc factor, and most cases the zinc ion of each finger is coordinated into the cysteine and two histamines. So whenever in the sequence there are cysteine and histamine, then the zinc ion, it attaches itself, it coordinates to that position with those amino acids and makes a zinc finger motif. The first zinc finger motif, the protein to be discovered, was TF111A and has 9 zinc fingers. It has attached with the 9 zinc fingers, and in this finger, the transcription factor that bind by means of a zinc finger motif has been demonstrated. Now, helix loop helix motif, that is the characteristic of the two helical segments which are prepared by intervening loop. And the proteins that are the basic there for BHLH motif always occur as a dimer. Example of transcription factor is, for example, BIO731. And the helix loop motif, that is containing transcription factor, play a key role in the differentiation of certain types of tissues, including skeletal muscle. This is their role in the transcription. Now, a transcription factor with a basic helix loop or helix motif has been demonstrated in this figure. And another third one is the leucine zipper motif. That is, the leucine occurs every seventh amino acid along a stretch of helix. And two helix of these types are capable of zipping together to form a coiled coil. A coiled coil protein, that is the made together the basic segment and the leucine zipper are referred to as a B-zip control. So these are the different motifs which control the transcription factor by a cell.